Hello lovelies, in this video the brilliant Dr. Edwards is going to be looking at one of the practicals that you'll do for your A-level biology. So we're going to be testing for glucose, and this involves making a standard solution and using a colorimeter and Benedict solution. So there are lots of things in here. We're going to be looking at the graphs, we're going to be looking at the maths involved, and this is one of the things in the exam they could ask you a lot of questions about that aren't necessarily exactly what you've done in class, so we're going to introduce you to some alternatives as well.
Hi everyone. Okay, so we're looking at the practical today that we can do to test for the presence of glucose in urine, and in theory test how much glucose is present in some urine, and we're going to do that using a calibration curve. So testing for glucose in urine is quite normal. If it's suspected that a person has diabetes, a doctor might request a sample of their urine and get it tested. In short, simple cases, they can use these little test strips that you might have seen before, where you dip the stick in and it will change colour depending on how much the concentration of glucose is present. And ideally it should be very low, so 0 to 0.8 millimolar concentration. And a blood test would then be used to confirm this, because obviously we need to double check that. And it could be caused by diabetes. It could also be caused by other things such as high blood pressure. If you think about high blood pressure, it means that you could be forcing more molecules out of the glomerulus, or there could be a barrier there. You could break those blood vessels. Or kidney failure, so the kidneys aren't working and they're not actually reabsorbing the glucose into the blood, and instead they're ending up in the urine. And so there's ways and means of doing it, and mostly it will be a blood test, and there will be other symptoms that the doctors will look out for. We can test urine on a quantitative level, so be able to actually confirm how much glucose is in urine, and we can do that using Benedict's reagent. Now there are two types, and it depends. You can use both for this practical. So qualitative Benedict's is the one you'll have used up until kind of GCSE level. So it produces that range of coloured precipitates when boiled, and it goes from like greens through to yellows, oranges, and then red. And that's when it's in the presence of reducing sugar. Obviously, the non-reducing sugar test is different, but we don't need that here because we're looking at glucose, and glucose is a reducing sugar. The other type of Benedict's is called quantitative Benedict's. It doesn't give that red precipitate on boiling. Instead, the amount of reducing sugar present is measured by the disappearance of the blue colour of the copper sulfate. You can use either. Some people say it's obviously easier and better to use the quantitative Benedict's if you're going to be using a colorimeter, which we are, because you don't have the precipitate, which means you don't have to have the filtering step, which can sometimes be slow and tricky. And so if you can get quantitative Benedict's, that's normally quite helpful for this test. Okay, so we've talked about that we're going to use a calibration curve. So what does that mean? What does a calibration curve mean? Well, your Benedict's can be used to test a range of known concentrations of glucose made using a serial dilution. So we make a serial dilution of a glucose solution, a stock solution, so that we know each of the concentrations in the tube. So I've got examples here where I've got 0 to 10 millimolar concentration. I know that in each tube, that's what was in there. Then I add my Benedict's, either type of Benedict's is fine. And then I run the test. So I put it in the hot water, get the Benedict's to work and create an action. And then I use a colorimeter to measure each of the samples. And that will give me an absorbance reading for each of the concentrations. I plot that, and that becomes my calibration curve. And we use a red filter because that's opposite to blue on the color wheel. And this makes sense. So you get a straight line, and it's still called a curve even if it's a straight line for each sample. So if we're using the quantitative Benedict's, so as we have more glucose, it gets less blue. You start and blank the colorimeter with your kind of 0, which is your blue Benedict's solution. So as you get less blue, you will have less absorbance because it'll be further away from that blue color, and therefore less glucose. And there's less color to absorb the light. That's why we say less absorbance. Whereas if we use the qualitative Benedict's solution, then you're going to go from blue, which is your start point, but we're not going to get less blue, we're going to get more red. So we're getting closer to red. So again, you start and you 0 your colorimeter with blue solution. And so we're getting less blue, but we're going more red this time. So the colour's actually getting sort of darker and closer to red, which will be on the maximum absorbance on our filter on the colorimeter. So as we have more glucose present, we're going to go from less blue to more red. And so we get a higher absorbance reading. So higher absorbance reading means more glucose is present in the solution. And that's because more glucose is present to have reacted with the copper sulfate to produce the precipitate. So we plotted our data to get our calibration curve, and once this is done, you can have a couple of unknown solutions, which will be a concentration of glucose that you're not sure what it is. You do exactly the same test, you do it exactly the same way as you tested your standard solutions, and you read the absorbance from the colorimeter. And then what you can do is you can use your calibration curve to then figure out what your concentration of your solution is. Because, for example, if you find out that your absorbance is 0.6 for an unknown solution, you can go across to your calibration curve, you can hit it, and then you go down to your x-axis, and then you can use that to tell you what the glucose concentration is. And here it's probably about 1.8. And then that's your estimate of your concentration of your unknown sample. Okay, so your equipment, and we're going to have a glucose stock solution, we're going to have distilled water, we're going to have our urine samples. I say that in inverted commas because we're not actually testing urine, we're testing 3 unknown samples of glucose, but we're going to label them as if they are urine samples from patients. We're going to use qualitative Benedicts today, but again, as I said, you could use either pipettes, test tube racks, and test tubes, colorimeter and cuvettes, and then a water bath set at 90 degrees, or like I'm using, we can use a kettle and some beakers. Okay, so the method then. We're going to make a sealed dilution of the glucose stock solution using distilled water. I'm going to show you a short video to remind you how to make sealed dilutions on the next slide. I will have just made that using my glucose stock solution and some distilled water to get a range of concentrations. You should make a record of the concentration that's in each tube by labelling it. Clearly label your test tubes for each patient as well, so you should have patient 1, patient 2, patient 3. These may have already been given to you. We're going to add 2 centimetres cubed of Benedict solution to each of the glucose samples, including the patient ones. We're going to place all the test tubes in the water bath together, leave for about 5 minutes, and then allow to cool down. We're going to then filter the samples to remove any precipitate. Use the 0 molar solution to blank the colorimeter using the red filter. Place a sample from each test tube into a cuvette and use the colorimeter to measure the absorbance and record it in a suitable table. Plot a graph of absorbance against the known concentrations of glucose to create a calibration curve. And then finally, use the graph to identify the glucose concentrations in the unknown patient samples. All right, so the hazards and sort of our risk assessment for this practical is pretty simple. Benedict's is an irritant, so it's got copper sulfate in it, which can be an irritant. So obviously wear safety goggles, wash skin if it comes into contact with the reagent at all, because it can irritate the skin. Obviously, don't ingest anything like that as well, but make sure you're wearing safety goggles, make sure you're just clearing up any spills, make sure you're rinsing your hands if any of it gets on your skin. Obviously, we're using hot water, about kind of 90-degree water, so making sure any beakers, if you're using, are not going to be transported while they're hot and moving around. Only half full beakers if you're using them to avoid any spills. And then any hot test tubes that have been in the 90-degree water, try and remove those with tongs rather than your hands. Don't ever hold a hot test tube. Those basic things we should hopefully be aware of. Okay, control variables just to keep in mind. Again, this is quite a simple one. There isn't much to think about in terms of technique because we're not thinking about having to keep anything really cold or having to have control variables, treatments or anything like that. So the same volume of glucose solution should be in each tube, in this case 2 centimetres cubed. Same volume of Benedict's added to each tube, again 2 centimetres cubed, and same length of time in the water bath for all solutions, and ideally they should go in together for 5 minutes. Come out together, cool down together, be filtered at the same time, etc. Just treat them all the same, your 3 unknown samples and your standard solutions as well. Okay, so a reminder on the next slide, what I'm going to do is give you a reminder of how to make a serial dilution. So this isn't exactly what I did, but this is just reminding you how to make a serial dilution. You will have to do this for yourself with your glucose solutions.
Here I'm doing a very bad thing. I've put food colouring in a solution. I hate it when people do this, but it is a really good way of showing colour to you. So I'm going to do a serial dilution down from here. This is a skill you need for biology and for chemistry. And the only reason I'm using red food colouring is so you can see the colours. There's 10 centimetres cubed of distilled water in each of these up to the line with the bottom of the meniscus. I'm then going to use a pipette to take 10 centimetres cubed out of here, put it into here, mix it, 10 centimetres from here into here, mix it, 10 centimetres cubed from here into here, mix it. So we're going down, concentration is going down by half every single time. I'm using a pipette to transfer the fluid. You'll see that the pipette also has a little line on it, and then once you've sucked it up to the line for transferring, just suck it up even further so you've got a gap at the bottom. We're going to start off with a concentrated solution in test tube 1. Actual numbers used here don't matter that much, this is just an example. But say that tube 1 is 40 centimetres cubed of 1.5 molar sucrose. Half of this volume move to tube number 2. Tube number 2 now contains 20 centimetres cubed of water. To that, we are going to add 20 centimetres cubed of 1.5 molar sucrose. Equal volumes. Give a final concentration of 0.75 molar sucrose. Through a bit of mixing, 20 centimetres cubed can be removed from tube number 2. This can be added to tube 3. To give a final concentration in tube 3, 0.375 molar sucrose. Made from 20 centimetres cubed of water and 20 centimetres cubed of 0.75 molar sucrose. Centimetres cubed, 0.357 molar sucrose, can be removed from tube 3. This can be added to tube 4. Give a 0.1875 molar concentration of sucrose. 80 centimetres cubed from tube 4, be moved to tube 5, added 20 centimetres cubed of water, give a final 0.01 molar solution. And please ignore all the colours, not the colours of writing, the colours of the liquids that I used on here. I know they're not really those colours, but it's just to help you see it and make it look pretty.
So once we've made our cereal dilutions, as we've looked at in the previous video, we're now going to actually get ready to do the Benedict's test on them. All I'm going to do is add 2 centimetres cubed of Benedict's to each sample before we put them in the hot water. Once I put it on, I'm going to give it a little mix for each one. Okay, so those are all my known samples, and then I have my patient 1, patient 2, and patient 3 unknown samples. So I'm going to add 2 centimetres cubed of Benedict's to those as well. So the samples have gone into the water bath, so I've used hot water from the kettle and just put those tubes in them, giving them kind of a shake and a mix, make sure they kind of evenly distributed through the hot water. Set the timer for about 5 minutes, and we're just leaving them to sit in there, waiting for the timer to go off. All the tubes have gone into the same temperature of water and have gone in for the same amount of time. So the next steps in the method are to blank the colorimeter, get it ready using a red filter, and we're going to use distilled water and Benedict's, and I'll explain that in the video. Also, to filter the samples first to remove any precipitate before they go into the cuvettes. And that's important because any precipitate left can make like a cloudy solution, and that stops light being able to transmit through the sample. So just using a filter funnel and some filter paper, filtering mostly the ones that have the precipitate. You can filter all the samples, and also just making sure that you don't overfill the cuvettes, but fill them up at least halfway. Okay, so we've filtered all of our samples, and now we need to measure them using the colorimeter. So here's my colorimeter, and it's a small one. It doesn't really matter what type of colorimeter you use, the kind of rules are still the same. So ideally, we probably want to be setting the wavelength to a red filter because most of our samples are blue. And if you're using the quantitative Benedict's, then you would definitely be using a red filter. You might find that some of the red-orange samples give funny readings using the red filter. So you might need to go somewhere between red and green, and that will just be something you kind of work out with the technicians that help you at school, or maybe through trial and error when you're taking your own readings. So I've set the filter here to green for now, and I might do a version with red as well. But first of all, we must blank the colorimeter, or calibrate, 0,
always the same. So what I'm using for that is I'm using my 0 molar sugar solution with Benedict's. So it's basically distilled water with the Benedict solution that we used. And then I'm going to put that in and press calibrate. So this zeroes the machine. This is our kind of 0 point. So any changes from this colour will give a different absorbance reading. So we're measuring this as a standard, and all of our samples we measured against this. So any change from this will be measured as an absorbance reading. Often, if you've used a calorimeter before, you may have blanked using just pure water. But because we're starting with a coloured solution, we're starting with Benedict's already is blue at the start, we have to make sure we use that as our standard, as our blank, because otherwise we would be blanking. Everything would have a really high absorbance reading because clear water would be very different from the blue Benedict's. Whereas we're looking for a colour change from the blue Benedict's, which is our start point. So that's why we blank with that in this case. And then once you've removed your blank, your colorimeter is ready to go. You can put your samples in. So this, for example, is my patient 1 sample. As an example, I put it in and it's going to give me a reading, so 256. And that's the absorbance. This here is to set the absorbance to transmission instead, so how much light, what percentage of light is transmitted through the sample, as opposed to how much light is absorbed by the sample. And sometimes different experiments will call for that if you're looking for how cloudy a sample is, for example. And then this is just to change the filter. So this is how we're going to do the readings. I'm going to do exactly the same thing for all of my standards and all of my 3 unknown samples, and then I'm going to use this data to produce my calibration curve.
So with all those readings that we took from the calorimeter, we have now plotted our graph of absorbance against the known concentrations, so my standards, and created this calibration curve. So now we need to use this graph to identify the glucose concentrations in the unknown patient samples. So patient 1, the absorbance was 0.256. So go and find that on your y-axis, draw a line across. Remember, always use a ruler so that you do not mess up with this. It's really good to make sure when you are asked to do this, whether it's in an exam or in your practical book, to make sure you actually do this on the graph so they can see that you've done this work. Sometimes it can get you marks in the exam as well. So that reading across to our line and then down to our x-axis gives us a concentration of 0.12. I would say this is not quite extrapolation, but obviously we had a 0, which is our blank, and then we started at 0.2 concentration. So it's a little bit outside our range of values we actually tested for this one because it's quite a low reading, but it's still acceptable, the method that we've chosen to measure it here. Second one, patient 2, was 0.571. So we go again up to our y-axis to 0.57-ish because we can't really do 3 decimal places here. Across to the line and down gives us 0.64. And then patient 3 is 0.810, so go up to 0.81, so halfway between that first box, halfway in that first box. Notice I've got 3 decimal places for my absorbance and 2 decimal places for my glucose concentration in the table. Could round up those absorbance values to get 2 decimal places across the whole table to make it neat, but because they're in the same column and they're the same decimal place number, that should be acceptable. So we've got across again and then down, and so this is going to be quite a high one, 0.96. Right, so those are our results. So what conclusions can we make from this? Because obviously the whole point of this practical is to practice doing the zero dilution, practice using a colorimeter, but then also using a calibration curve to find concentrations of unknown samples. So normal urine glucose is about 0 to 0.8 millimolar. So what does this tell us? It's probably that patient 1 and patient 2 have quite normal range of urine glucose, whereas patient 3 is definitely outside of that range and definitely higher than 0.8. So in conclusion, we'd probably suggest that patient 3 has quite high urine glucose levels, and that could indicate diabetes. So they should be given a fasting glucose blood test in order to actually confirm that, as obviously well as looking at some other samples, symptoms as well. Okay, so that is this practical done. Hopefully that makes sense. Hopefully we've covered some tips for doing the practical, but also calculating your results and writing up your kind of findings from this practical as well.
Ouch! This is why in some videos I explain scratches.